the first week we covered how the eye works, and the eye is part of the cortex, has the same layered structures of the cortex, and people are now starting to use looking at the back of the eye, which is the retina, to actually measure the state of your brain. They can tell if you're potentially going to get dementia or Alzheimer's or Parkinson's by looking at the health of the cells in your retina, because they in some ways mimic the cells in your cortex. We then went on to see how the visual cortex works, and then how what areas combine the information from the visual cortex to be able to perceive objects and extract them to the background. We then went to see how motion worked, and how special areas, again, produce your sense of motion. And today we'll see how the rest of the brain works. So in one short hour, we'll cover the rest of the stuff. So this is sort of the essential parts of the cortex. It has 4 anatomical subdivisions. We studied this part here. It's called the occipital lobe, and it is where V1, V2, V3, LOR. Then over here, on the side and underneath, is the temporal lobe, and in the underneath part was where we found the fusiform face area. Over here on top is something called the parietal lobe, and that's, you might recall, it's part of that action stream, and we'll study it in more detail next week. And finally here, we have the frontal lobe, and this is probably the most important part of your brain in terms of it allows you to make decisions and decide what to place into your long-term memories and what not. Now, the important landmarks are this valley down here called the central sulcus, and it's the division line between the frontal and the parietal cortex. And over here, something called the lateral sulcus or sylvian fissure. It's called a fissure because it's actually deeper than in any of the other sulci. This thing on the side here is like, you can imagine it's like a chicken wing coming off your brain. So this part here is not really attached to the brain, it's just on the side. It's underneath here, and on top that the 2 cortices are attached. And there's down here is where your auditory system is, mostly deep down. So now information flows from the primary sensory areas to the higher sensory areas, then to the association areas, then to things called the premotor areas, and finally to areas that actually produce movement. So you can think of it as a loop. You perceive something and you act on it. Now we saw here, back here, the visual areas, primary visual cortex. That's the first where the eye through the lateral geniculate first hits the brain. Over here, just behind the central sulcus, is something called the somatosensory cortex, and it is where touch first enters the brain. So everything you feel from your skin, and also taste and the vestibular information that's coming from your balance sensors in your ears. And over here, also near the ears, is the auditory system on the temporal lobe. Now, from the primary, information flows, we saw in here, in the case of the visual areas, to V2, V3. Similarly, from the primary somatosensory cortex, it goes to higher somatosensory areas, and as well from the primary auditory cortex to higher areas. From there, it goes to the structures that we'll talk about here today. And those are the association areas. And there are two, the prefrontal and something called the parietal temporal occipital, because it covers both the parietal, the temporal, and some of the occipital. PTO for short. And from here, information flows to the premotor areas, and finally to the motor area, the primary motor cortex. And then from there descends down the spinal cord, and you produce movement. And interesting here that this is just in front of your central sulcus, and your sense of touch is just behind the central sulcus. And we'll see, just like the eye is mapped in the primary visual cortex, so the fovea is mapped in a certain part of it, and the periphery another part of it. So all your body parts are mapped in your primary sensory areas, so your fingers and the skin on your back and the leg. And the same thing here. Which muscles you're going to contract are mapped in these primary motor areas. Now, you can think of everything that happens in your brain as a reflex. And some reflexes are complex and some simple. So something simple would be a fly lands on your hand and you swat it. It's a very simple reflex. And something like writing down the name of an object that you see is far more complex. And we'll look at the more complex pathways in the lecture today. Now, we saw before that we have this black stuff is your skull, so it's bone. But underneath this black stuff is gray stuff. It's your gray matter. And you can see that it has lots of folds. And it's because the gray matter has been crumpled to get as much of this gray matter into your cortex as possible. Because that's where all your cells are, all your connections lie, and the connections are what forms the memory. So everything you remember is located in this gray matter. And you have something like 100 billion of these neurons in your gray matter. Now, in the white matter is where the connections lie, the axons from one area to another area. And we saw, for example, that from V1 you went to V2, and from V1 you also went to area MT, MT, and also you went from V1 across the corpus callosum to a neuron on the opposite side. So these are all the connections that get formed. So the whole brain is extensively interconnected. And if, for example, any one part starts firing incorrectly, that it becomes too hypersensitive, you can develop epilepsy. And that epileptic fit will quickly spread to all parts of your brain. Now, having 100 billion neurons is not a big thing. Most laptops have memories that exceed that in terms of how much information they can store. But what differentiates a laptop from your brain is the connections. And we have these neurons make something like 100 trillion connections. So 1,000 times more than the number of cells you have. So each neuron connects onto 1,000 other neurons. So not just 3 lines here, but imagine 1,000 lines for each one. And that's what makes the brain different. Now, we saw before that gray matter requires oxygen, and the heart can only pump so much oxygen into the brain, so the brain becomes very efficient in how it utilizes oxygen and where it goes to. So it just pumps oxygen to those areas that are being activated at one time. And we saw in the first lecture that those areas are kept to be a small number just to not use up too much oxygen. And that's lucky for us because you can the oxygenated blood is easily measured using fMRI, and we can measure those parts of the brain that are receiving lots of oxygenated blood, and then see what parts of the brain are involved in the task. And that's called functional fMRI, magnetic resonance imaging. And that's a powerful new tool that was developed, a little more than 10 years ago. And it's exploded. There are, it used to be maybe 100 articles were published a year. Now there's something like 100,000 articles are published a year. Western has become one of the key centers for doing functional MRI research. So if you are in any ways interested in graduate work, go visit the Brain and Mind Center. So connectivity is important because of speed, you know. So each neuron connects to 1,000 others, and parenthetically, these connections are strong because you make up the 10 synapses just to make sure on each neuron. And in contrast, most PCs and Macs have only 64 connections. So it's a big difference. And but yet no computer, as far as I know, can understand a sentence like a rope is for pulling, not pushing. And a young child can understand that. They estimate that if one tried to pack everything that's in any one of your heads into and simulate it on a computer, they'd require the complete internet of the world to get close. Okay, so let me try to. Here's a question. Again, we'll do a pop quiz. So you raise your finger if you think the answer is this on the first one on top. 2 fingers for the second one. 3 fingers for this one. 4 fingers for this one. Okay, and I'll say when, and you guys have to raise your hand at the same time. The question is, suppose you're using Twitter, and everyone in the world also used Twitter, and each tweet connected to 1,000 others, and they in turn to 1,000 others. How many people could you reach? How many tweets would it take to get everyone in the world? So, 1, 2, 3, now. Go on, raise your hands. Try to guess. There's one with 4, one with 2. What about the others? 3, 2. That's 3. Okay, 3, 3, 3. The answer is, let's see. Yes. So 1,000 times 1,000 times 1,000. It is with 4 you reach 1 trillion. So that's we're at 7 billion at the moment. So you'd have a lot to spare at a trillion. So you can imagine the power of that in your brain. So with 4 connections, one synapse will talk to another synapse, will talk to another synapse, talk to another synapse, you'll have reached every synapse in your brain. Okay. So information quickly spreads. And it is what makes the difference. And you can see this wiring. One of the other new things with MRI is you can not only image the cells, but you could image the wiring. And here's a picture that you get with the wiring. And in the States, they're creating this human connectome, something like the big genome project, where they're gonna figure out what the connections are, every connection in the human brain. And if you click on this at home, you'll see some of the pictures they're getting. They're truly amazing. Okay, we saw before that we had 6 layers, and they're anatomically distinct. We have in some parts of the brain, one layer will be thicker than others, and that's because it depends how information flows. So it always comes into layer 4. It always goes out from layers 3 and 5. Now we saw that V1 had a very thick layer 4, so it was a striate cortex, and area the areas that produce movement in the motor cortex, they are very thick layers 3 and 5 because there's a lot of information flowing down your spinal cord to produce movements. Now, how's the brain organized? There were several theories put forward, one by Lashley, which largely based on studies in which small parts of the brain were removed from mice. And he found that he thought that maybe the brain was a pretty uniform structure, that if you took out, when he took out a small part, very little went wrong. And on the other hand, when Hubel started doing his experiments, he found these simple and then these complex cells, and these hypercomplex cells. And somebody had the idea that, well, maybe when you keep going that way and eventually have a cell in the brain that recognizes just grandma. Now, so the evidence for is that some lesion will impair, we saw in the fused photo face area, selective loss of face recognition. But the evidence against it too is that it's not a single cell, but it's a group of cells. Because rarely do we have wake up the cells are dying every day in your brain. And luckily, they're also being replaced, we recently found. It used to be thought that at your age, that's as many cells as you probably get ever in your life. But it looks like we keep getting more, so don't worry about the odd one kicking out. But anyways, cells die, and you don't wake up one morning not being able to recognize grandma just because one of your grandmother's cells died. There are many cells that represent the perception of grandma. And the evidence here for this theory was that a small lesion will generally be hard to detect. Evidence against it is that, as we saw in the preamble to this lecture, different parts of the brain do different things. There is a visual cortex. There is a somatosensory cortex. So the brain is not homogeneous. So it's not a single cell and it's not homogeneous. It's somewhere between these two extremes. Now, is your cortex, everyone's cortex here in the room, the same? And the answer is no. The cortex is very plastic, and depending on what environment you grow up in, and what damage might incur as you develop, changes will occur that will differentiate your brain from other brains. And of course, there's all the genetics that everything that you inherit from your parents and grandparents and everyone else before that. We saw that if an input is missing, some other part of the brain takes over that area. So if one eye is slightly impaired, the other eye will take it over. Well, the same thing occurs if let's say, both eyes are missing and you're blind, and that area then isn't just silent, but it starts becoming active to things like speech and the auditory system. And also, for example, language. If early in life your language centers on the left side are for some reason damaged, then the ones on the right take over and your language function remains entirely normal. Now, but there are differences in the brain. It's not quite as extreme as we thought maybe 20, 30 years ago, but there are differences. Language is usually on your left cortex, and that's called the dominant cortex, largely because language is an important function. So when the neurosurgeons do surgery, they're especially careful to avoid the language areas of the brain, and for that reason, this side has been called the dominant side because of the importance of language. But it's also involved in reading, writing, and things that are analytic, like algebra. So rule-based. So A equals B and B equals C. We know that A equals C. So those kind of rules. The other side here, the non-dominant side, and usually the right, is more involved in things that involve parallel processing. So, for example, to recognize the face, you need to do a lot of parallel processing. You see the whole face as a unit, and in seeing that unit, you automatically recognize how close the eyes are, and whether or not certain features are there or not, and instantly recognize that this person is familiar or not familiar. But also, because of that, it's very useful in things like geometry. So we in geometry, one triangle resembles another triangle. You can see here does things like C looks like an O, or an I resembles an L. And music is another important function. But we saw that even the fusiform face area, there's a fusiform face area on one side of the brain and also on the other side of the brain. The one on the right is usually bigger and more important in most people. And then the one on the left, we saw that parts of it were being taken over by language. Now, what happens when you cut the connections between the two halves? So each part is doing something different. but what if they become disconnected? And uh, the, one of the ways people have been able to study that is patients that have severe epilepsy. One way of controlling that epilepsy is to disconnect the two halves of the brain, and that controls the epilepsy in some ways. At least it can't spread to the opposite side. And in studying these brains, you see interesting things occur. So you can, for example, have the patient look straight ahead, show a picture in his left visual field. That will go to the right visual cortex. Now, he can then pick up the apple, because his left arm is controlled by his right cortex. He can recognize that this is an apple, at least this side of the cortex can. But the person can't say the word apple, okay, because the language side isn't getting that information. And interesting, if you talk to these patients, I think I might have a link to some of those patients in this week's links section, and they operate quite normally. They're like normal human beings, except that sometimes the two sides are having conflicting sort of sentiments. One that I read about, he would hug his wife with one hand and push her away with the other. So one side and the other side had completely different feelings. An odd thing to imagine, having two parts of you in one head. Now, the advantage of lateralization is that the corpus callosum forms a very long pathway, and by confining all your connections to just one hemisphere, the connections become shorter, and that improves the speed. This duplication would be a waste in some ways. One can have double one's capacity. One side of the brain can be doing one task, and the other side of the brain can be doing another task. The disadvantage of this is that there's less redundancy. Okay, so one part gets damaged, the other half can't take over, as sometimes happens in early damage to the language function. Okay, so that's a survey. Now let's get down to the two association areas that we talked about. What do they do? Well, the first one let's talk about is the prefrontal association area. This is here in front of the central sulcus, and it's getting larger and larger and larger. It's what largely distinguishes us from every other species. Well, not every other species, most other species, because dolphins, whales have pretty big frontal lobes. Now, just to test your frontal lobes, one of the things that the frontal lobe excels is something called working memory. And one example is spatial working memory. If I close my eyes, I can still remember where this mouse is, and I can still move it around. Okay? And you can do the same. Whoops, and I lost it. What did I do? You know what I pressed to get here? I thought I did. Okay. All the way to here. Okay. There we go. Okay, back to here. Hopefully I will not close my eyes and get back to here. Okay, so let's test your working memory. Here you see an egg. We'll see two chickens descend, and then you have to remember where the egg lies. Okay, does it lie here or lie here? Everyone that thinks it's here, hit the tables. Everyone who thinks it's here, hit the table. Okay, thank you. All of you have good working memory. Working memory takes a few years to get started. One of the things that you can look at if you have young nieces and nephews that are about 1 year old is how easy it is for them to get distracted. If you have a toy and you cover it with something, it doesn't exist for them. So out of sight becomes out of mind. So working memory, so in a sense, there are cells in your frontal lobe that do this. Okay, so nothing's happening here. An egg appears, it starts firing. But you notice when the egg disappears, it continues firing. And then the egg appears over here, it doesn't cause it to fire. So it depends where the egg appears for this particular cell. And other cells will code other locations. Now, one of the important chemicals or transmitters in the frontal lobe is the dopamine. And without dopamine, you get depletion of working memory. And this dopamine originates from a structure called the basal ganglia. And Parkinson patients have a lot of trouble remembering targets. If in sight, if you could see something, then it's there. If it disappears, it's gone. So I don't know. There was a movie a long time ago where the doctor who discovered this dopamine was involved, and one scene in the movie, he takes these patients which are rather catatonic. So he stands them all up in a row, 6 on one side, 6 on the other, and they're standing there almost like robots, without any actions and without any emotions. And then he throws a basketball between them, and they just bounce the basketball back and forth between the 12 people standing there. No trouble at all. But because they don't have working memory, they can't initiate their own movements. So they can't act on something that isn't there. If something is there, they see it, they react to it. If it's not there, they can't compel it to appear. So this is involved in two very important. With working memory, you can make decisions and you can plan. You can play chess games. You can imagine what the next move might be and devise a plan. Now, one of the things that happens is that it's also a cause of frustrations. So if you have a plan and it doesn't succeed, you get frustrated. If, however, you don't have a plan, you have no need to be frustrated. And they discovered that if you did something called frontal lobotomies, it has had a calming effect on patients that used to be aggressive, but it also destroyed initiative. Now you might think that this is a horrible thing to do, a frontal lobotomy, but the fellow who discovered this, Dr. Moniz, he won a Nobel Prize for discovering this technique. And there's a fellow here, shown in this picture, Dr. Walter Friedman, who was very popular in the States as somebody who did this surgery. He did thousands of these surgeries. And he'd do them largely, from his office. And here you can see, let me just forget about this mouse and work on this one. You can see here, he's tapping this sort of ice pick through the back of the eye. So it goes behind the eye, and he just taps into the cortex, because the bone is relatively weak there, and it just squishes this ice pick around and cuts the fibers to the frontal lobe that way. And he used to do thousands like that all over the States. And you think that maybe, some uninformed people would allow this to happen to their relatives and friends, but Rosemary Kennedy, this happened to Rosemary Kennedy, the sister of John F. Kennedy. I mention these things because things that seem to, these are all medical students watching this happen. And maybe someday you'll be a medical student, and hopefully you don't have to watch something like this happen. But I'm sure there will be things that you'll be taught that 20 years later we say, oh my God, why did we ever do this? Anyways, let's move on to the next part of the brain, the PTO. It's a huge part of the brain. And it does many things. It's where we have polymodal convergence. It's a big thing, but it basically means that this is a great place for combining all your senses. We saw that vision is here, touch is here, the auditory system is here. So if we want to do something that depended on all three things, this is a place to do it. It's also a good place in which to do something like at your attention. I'm going to just turn this guy off so he doesn't keep interrupting the system. So we'll see in a moment how you could why attention can be important in many functions. And it's also where memory lies, especially this inferior part, and memory of faces, but of most things that we recall. And we'll look more at memory in the last session, lecture 12. Now let's look first at attention. Okay? They did an interesting experiment in this university, Western Washington University. They went to something like the UCC Square and had one of the students ride this motorcycle around dressed as a clown. And they questioned the people walking by if they had seen anything strange. And half the people that were talking on cell phones didn't see the clown. And when they test drivers or do tests in simulations, they find that when you're texting and driving, you're worse than being drunk. So this is because when we'll see in a few moments, attention is limited. And it's limited for an important reason. We don't want the whole brain active. We just want to have our attention focused to the important parts. And this is this the fact that you have blindness when you don't have attention means that it's true when you're driving a car and you're listening to this lecture. If you don't attend to what you're hearing, you won't see it. To give you an example, raise your hands without saying what it is, but when you see what's changing in this picture? There's something changing. 1, 2, 3, 4, 5, 6, 7, 8. About half the class. Come on, we're still some more students. It's easy. It's easy. Okay. Notice how when your attention is focused on it, it's obvious. Okay. How could you have missed it? You say to yourself. And so a fellow called Resnick did a lot of these, and there's a whole bunch of links again in the section on links that shows you all kinds of things, pictures that flash like that, and you're amazed at how you don't see things that are changing. So, an analogy of attention is like a flashlight, and your eye and your visual cortex sees all of this, but it doesn't pass further down into the dissociation areas without attention. So, a very famous scientist called Kutel, he came up with the reason for this was that your brain would fry, just too much energy. And of course, we know that he's wrong. What you'd do if you lit up all your cortex, you would faint. Much the same effect, but a different reason. So how does attention work? You can see here working memory up here, so you can think of your associations up here. You can think of your visual areas down here, and this is the PTO, the attention part of your brain. So something happens, it attracts the attention, and while it's flowing through slowly, other things can happen over here, or even over here, and you don't notice them happening because it's all being consumed. All your attention is being consumed where this flashlight is on, and during the process time, it can't even see what's new happening here. Now, normally, things that are different over here attract your attention. So if I had these are vertical lines and these are oblique lines, and one color and different color. So this stands out. If you're looking at a visual field full of these things, this will stand out, and it will attract your attention, and that will get down to your working memory. Sometimes what you do is focus your attention onto different objects. So they appear and you go through it one by one. When you're reading, you're basically doing that. You direct your attention to the first word, then the second word, then the third word. And normally, you're not blind to this bicycle here that's flashing. What I did before was a trick. I had one scene, then a flash of a white, and then the other scene. But if I put them back to back without the intervening white scene, you can quickly see this bicycle appear, and just its appearance, this thing happening out in your periphery, will attract your attention. Just like if one of you stood up in the class and suddenly waved their hands, everyone would look at that person. Now, without that standing up and flashing away, it takes time. Okay, so can you see where Waldo is? It'll take time for you to figure out which one of the guys is Waldo. Okay, I think this is Waldo. I'm not sure, but my guess is this. I've studied this for a while, and that's the only Waldo I think I can see. So why does it take time to find Waldo? Well, there's 3 reasons for it. First of all, Waldo doesn't jump out of the street. You've got people with similar outfits, similar colors, and he blends in. Secondly, it takes time to shift your attention from one object to another. And you can shift your attention either of 2 ways. One is called covertly, that is without moving your eyes, and the other is overtly, with moving your eyes. So without moving your eyes, your brain can look all around where you're staring and decide, well, maybe that's Waldo. And then you'd make an eye movement to that point and say, oh, is that in fact Waldo? And then finally, it takes time when your eyes are actually, your focus is looking at Waldo, to decide, yes, indeed, those are the characteristics of Waldo. Now, so attention is not the same thing as arousal. Your general level of arousal is affected by things like caffeine. When you have a lot of caffeine, you have arousal. And this is mediated by structures like the locus coeruleus, which has like a neuron that goes to everywhere in the brain and makes tons and tons of synapses, sort of waking the whole brain up. But it's not something that's focused to small areas like attention. Okay, some examples of attention. This will involve you doing this again. So, you saw there something flashing and then it appearing, okay? So I want you to do that. So a square will appear, and then don't hit the table for the square. But then when this circle appears, the little yellow circle, hit the table as fast as you can. But you did very well. But there was a different emphasis on how many hit the table if you were listening to the sounds. So if you're like most people, that last one, and you could hear it from the audience, that was a very that circle went up at a very unexpected place. And then two trials back, you also were in the wrong place. But then in most of the other trials, the square signaled where the circle would appear. And in those trials, you were much faster because the attention was focused at that area and you reacted quicker. So that's an example of spatially directed attention. Now we'll do some examples of attention that's on directed at other things. The first one is this. So again, these objects will appear over here, and you have to click the mouse when two objects in succession are of the same color. Okay? So we'll start now. Again, get ready to hit the table. Okay. So when you did that, some parts of the brain became active. And those were the, whoops. The areas that were looking at color, and those were sort of located back here in the cortex, related to the double opponent cells and where they project. Now, in another experiment, experiment 2, when two successive objects have the same shape, okay, so now structures, you can imagine that LOC would have been very important. So you directed blood flow to LOC. And lastly, in the same direction. So we'll move twice in the same direction. Okay. So people, there was a paper published about 5 years ago that did exactly this experiment. And for example, in this last one, blood flow was very marked here, up here, where the interparietal sulcus, not the interparietal, the inferior temporal sulcus ends up, so in area MT. Okay, final test. Can you see the odd line? Easy. It just pops out, eh? So you see a bunch of horizontal lines, and this odd line pops up. So your attention easily shifted to it. Let's make it a little more harder. So now it'll be a little harder. Again, hit the table when you think you've spotted the odd line. A little harder. Now, can I? Is it this one? Yeah. If you sort of, now that you know it's a black one, you can sort of tune your brain to black. You know, it's going to be a black line, and again it pops out. So if you know what color it is, it pops out at you. So this time I'll show you a different bunch of lines, but again, look for the one that's black. See how much quicker it was when you tend to a particular type of line, a different so color can direct your attention. So you're looking for a line of a particular orientation, but being able to detect color can speed things up. So we saw there are two types of things: spatial, like a spotlight; feature-based, whether it was orientation, whether it was color, sort of attributes. We can also attention being directed by objects. So over here, these lines, this thing pops up automatically, but the letter A here pops out if you see all these numbers versus a letter. So it's a differentiation in the type of line. And over here, again, this object pops out because of the different thicknesses of line. Okay, so we have attention. The opposite side of attention is neglect. Now, neglect is simply it's lack of awareness. Now, I'll show you again in the links how if you don't have attention and you neglect, you can have half the portrait in this figure. So we'll see how a patient that had this type of neglect fared. Now, neglect, if you have a right PTO lesion, is different from the lesion that what you'd have if you have a right V1 lesion. So if you're looking here at the X, if this face is over here, you see it. If this face is in your blind spot, blind cortex, you don't see it. So the right V1 is missing here, so you don't see everything to your left visual field. The right PTO is different. The right PTO, if attention is focused to an object, in a sense, forms a frame around this object. And in this case, the left half, because it's a right PTO, is not visible, independent of where you're looking. Okay? So if you're looking here, the left half is missing for the right PTO, independent where that object lies. Now, if you have the opposite lesion, the one on the other PTO, you don't have the same problem. Why is that? Well, some people have suggested that in the parietal cortex, you have on the right both visual fields represented, and on the left only one, the right visual field. So you can imagine that if you have a lesion here on the left PTO, this side is missing here, but it's still represented here. Okay? In contrast, when you have a right PTO lesion, then the left visual field is always affected. Coordinate frames. That's the last thing I'd like to cover. Now, there are two ways of representing object locations. One is allocentric. So that's what allocentric is. The nose is in the middle of the face. The eyes are above the nose. So you can imagine that there's a table and there's an apple on the table, and so you've got this measuring stick on both sides of the table, and you can locate this apple on this table. Another way of saying where things are is egocentric. So in this case, it's not where the apple is relative to the table, but where the apple is relative to yourself. And again, in this case, the grid is you, and that you can be many things. It can be your body, your head, or your eye. And we'll look at those various possibilities in next week's lecture. Now, the best way to appreciate these differences is to imagine this is of transparency with you in the center of it, and this is a transparency with objects, different objects. And you can place the two together. If it's allocentric, okay, then this is, for example, the room, and you're walking around the room, and so you are moving with respect to the objects. All these objects are staying still. If it's egocentric, then you're staying still, and all these objects are moving with respect to you. And then we can have sort of nested frames, so that you have let's say, a room and a table and an apple on a table. And when you move this table, everything that is going on with the apple also moves. Now, why is that important? Well, it's important because these two streams, the what ventral stream and the where dorsal stream, use these different frames of reference. This one here uses a me-centered reference frame or an egocentric reference frame. This one down here uses the allocentric or object-centered reference frame. And you can see that that makes sense. You want to code a face in terms of an object. You know, the nose is here with respect to the face, whereas here you're doing actions. So you want to reach towards actions. These actions are relative to you. So if you want to walk to a restaurant, you use the where stream because you want to determine where you want to go. In contrast, where you want to recognize the faces, like I said, the features of the face are depicted on it with respect to the face. And that's it.